Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing DBT JRF entrance exam question paper conducted in the year 2015. I have divided the question paper into two parts and this is the part 1. So coming to the first question. The molecular weight of protein is 30 kDa. The minimal length of mRNA encoding this polypeptide will be close to. So molecular weight of one amino acid is 110 daltons. And number of amino acids in 30 kDa or 3000 dalton is 30,000 divided by 10. That is 272.72 amino acid. Now one amino acid, it is coded by 3 nucleotides. It is not codons. I made a mistake there. It is 3 nucleotides. So 272.72 amino acids will be coded by 818 or approximately 800. So the answer is option A. Next one. What is the amount of protein required to prepare 5 ml of 1 micromolar solution? Molecular weight of protein is 25 kDa. So volume is 5 ml, molarity is 1 micromolar and molecular weight is 25 kDa or 25 into 1000 daltons. Substitute in the formula. Molarity is equal to 1000 Wb by Mb into V. Wb is the weight of the protein required and Mb is the molecular weight. So substitute in the formula and you will get 125 micrograms, or option 1 is the correct answer. Then next one, calculate the concentration of NaDH solution whose A340 nanometer is 0.8 OD. Optical path length is 10 millimolar. NaDH molar extinction coefficient is 6220. So you know the formula. A is equal to epsilon Cl, where A is the absorbance, epsilon is the molar extinction coefficient, C is the concentration, L is the path length. And path length they have given as 10 millimeter, convert it into centimeter. So it becomes 1 centimeter. Now substitute in the formula and you will get 128.6 micromolar, or option 1 is the answer. Then next one. The data below are from measles and stall experiment. Which model of DNA replication is supported by the results shown in the table below? So in the generation 1 there is no hybrid DNA. In generation 2 also hybrid DNA is not there. 3, 4 also hybrid DNA is not found. The DNA replication, as we know, it is semi-conservative mode of replication. If it was semi-conservative, then you should have got some amount of hybrid DNA also. But here no hybrid DNA is found. So it is naturally conservative. DNA replication is conservative. Answer is option 2. And according to conservative model, the entire original DNA double helix serve as template for a new double helix such that each round of cell division produces one daughter cell with a completely new DNA double helix and another daughter cell with a completely intact old or original DNA double helix. That is conservative model of replication. Th1 response is characterized by the secretion of which of the following combination of cytokines? They have asked for Th1, that is interferon gamma and interleukin 12. That will be the secretions by Th1 cells. Then next one. In hybridoma production, aminopterin is added after fusion 2. So after fusion, it causes the death of unfused myeloma cells. Answer is option 3. Then an alpha helical conformation of a globular protein can be determined by. So this can be determined by circular dichorism. Answer is option 4. So coming to the next question. Athymic nude mice, NUNU, are difficult to breed and maintain because the homozygous NUNU females are sterile. How will you propagate it to get maximum nude mice progeny? So first option, by mating normal males to heterozygous NU plus females. So these are the normal males and this is NU plus female. You don't get any NU mice progeny over here. Then option 2, by mating homozygous NU males to heterozygous NU plus female. There you get 50% NU mice progeny. But they have told that you should get maximum nude mice progeny, so we have to see for other options also. Then by mating heterozygous NU plus males to heterozygous NU plus females. There also you get NU progeny, but it is only 25%. The next is by mating of heterozygous NU plus males to normal females. So NU plus into plus plus, that is normal female, so you will not get any NU mice progeny. So out of option 2 and option 3, which are the options where you get NU progeny? Option 2 is having 50% NU progeny and option 3 is having only 25%. So the answer here will be option 2. Then next one. The base sequence of a short piece of DNA is A, G, C, T, T, A, C, G. During replication, a transition mutation occurs in the complementary strand synthesized on this piece of DNA. Which of the following is mutated complementary strand? So they have given some complementary strands here. We have to see which one out of these is mutated one. So here A, G, C, T, T, A, C, G, if it is the option 1, that is T, C, G, A, T, C, G, you can see that purine is replaced by pyrimidine because it is not a transition mutation. And transition mutation is whereby a purine is replaced by a purine and or a pyrimidine is replaced by a pyrimidine. So in the option 1, none of the cases is a purine replaced by a purine or a pyrimidine replaced by a pyrimidine. Similarly in option 2 and option 4 also. But if you see option 3, you can see that here in this region where I have put it in the box, there is a purine being replaced by a purine. So option 3 is the right answer. Answer is option 3. Then coming to the next question. Uncoupling of LDL receptors with their ligands occur at which of the following compartments? So in receptor mediated endocytosis, the macromolecule, that is LDL, to be internalized first bind to specific cell surface receptors. Then these receptors are concentrated in specialized region of the plasma called clathrin coated pits. These pits bud from the membrane to form small clathrin coated vesicles containing the receptors and they are bound to macromolecule, that is LDL. The clathrin coated vesicles then fuse with early endosome in which their LDL contents are sorted for transport to lysosome or recycling to plasma membrane. And in case of LDL, the receptor is then recycled to the plasma membrane while LDL is transported to lysosome. Whereas where cholesterol is released for use by the cell. So what have they asked? Uncoupling of LDL receptors with their ligands occur at which of the following compartments? So it is in the early endosome because the clathrin coated vesicles fuse with the early endosome in which their LDL contents are sorted for transport to lysosome or recycling to the plasma membrane. So the answer is option 3. Then next one. Transport of cargo from nucleus to cytoplasm through nuclear pore is regulated by RAN-GT pace. Answer is option 4. Which one of the following is cobalt containing vitamin? Of course it is vitamin B12. Answer is 4. Brifildin A inhibits protein transport from. So Brifildin A inhibits protein transport from the endoplasmic reticulum to the Golgi indirectly by preventing association of COP1 coat to the Golgi membrane. So answer is option 1, that is endoplasmic reticulum to Golgi apparatus. Which of the following diseases is caused by bacteria? Measles is caused by virus, tetanus is caused by bacteria, and Mayer's disease is caused by herpes virus, and mumps is caused by virus. So the answer is option 2, that is tetanus. Then next one. Which of the following antibiotics is used to demonstrate the new fresh protein synthesis in response to an inducer upon induction in a microbial system? This is a repeated question. I think it is taken from 2013 DBT paper. I have done that. You can go through that. So the answer is chloramphenicol. Tetracycline, it inhibits the initiation of protein synthesis itself. And chloramphenicol, it inhibits bacterial protein synthesis by binding to the 50S ribosomal subunit. So chloramphenicol will be the suitable antibiotic. Then next one. And carbencillin and ampicillin, they interfere with the cell wall synthesis in bacteria. Then next one. Both somatic hypermutation and isotype switching depend upon a highly specific enzyme of adaptive immunity that is made only by B cells proliferating in response to antigen. The name of the enzyme is, so it is activation induced cytidine deaminase. Answer is option 2. Then AGO proteins are associated with. So AGO constitute the core of the RISCs, that is RNA induced silencing complex, with different members having variety of protein binding partners and biochemical properties. So RISC uses the siRNA or miRNA as template for recognizing the complementary mRNA. When it finds a complementary strand, it activates RNase and cleaves the RNA. So AGO protein is associated with RNAi effector complex. The fusion between protoplast can be enhanced by subjecting them to high electric current. Answer is option 3. The next one. If a nucleotide sequence encoding a protein is known and a homologous protein to be identified, which of the following will be the best analysis tool? So BlastX, it searches the protein database using translated nucleotide query. So answer will be BlastX. The next one. The concentration of which of the following plays an important role in somatic embryogenesis? It is PO4 3-. Option 4. Then simple sequence repeats markers are derived from. They are derived from both coding and non-coding sequences. Which of the following enzymes are required for making plant protoplast? So cellulase and pectinase. Answer is option 2. The floral dip method is commonly used for genetic transformation, where you dip the flowers in agrobacterium suspension for a particular period of time. The next one. Rate of centrifugation of a particle in a centrifuge is increased by increasing the centrifuge speed. The next one. Which of the following is true during the separation of biomolecules by reverse phase chromatography? So reverse phase chromatography includes any chromatographic method that uses a hydrophobic stationary phase. Mobile phase solvents should be miscible with water. So mobile phase is hydrophilic and stationary phase is hydrophobic. So stationary phase is less polar than the mobile phase. Yes, that is correct. Identify the incorrect match. Koch germ theory, Pasteur blood clotting theory. That is incorrect. The next one. The first smallpox vaccine is an example of. It is an example of live vaccine. Answer is option 3. ELISpot assay is traditionally used for measuring. So Ellie spot assay is a measure of the frequency of cytokine secreting cells at the single cell level. So it should be cytokine secreting cell. So frequency of T cell response. Yes, that is correct. Ellie spot assay is used traditionally for measuring frequency of T cell responses. Then next one. Fertility factor is related to conjugation in the same way as bacteriophage P1 to transduction. Answer is option 2. Hemorrhage is the brain tissue with loss of consciousness is known as apoplexy. Answer is 4. Which one of the following methods is frequently used to create transgenic animals? Nuclear microinjection. Answer is option 2. Which of the following is an atypical signaling receptor? So mannose receptor. It is an atypical signaling receptor. The critical regulatory circuit in the circuit of emotions is amygdala. Answer is option 3. Which glial cells participate in the reuptake mechanism of neurotransmitter from the synaptic cleft? So it is astroglia. Then red data book contains data of threatened species. Conservation within natural habitat is in situ conservation. MAB program stands for Man and Biosphere. Marine organisms that require oxygen levels typically in the range of 2 to 10% for growth would be classified under microaerophils. Along which plate boundaries do the majority of the world's earthquakes occur? So it is the convergent. Answer is option 3. Which of the following will increase the salinity of a particular area of coastal water? So freezing of water. Answer is option 4. An individual has the genotype. They have given the genotype. Assuming independent assortment, what frequency of gametes will have the genotype A, B, C, D, E? All small letters. So they have asked for the frequency of gametes. So there is a formula for total gametes, that is 2 raised to n. So 2 raised where n is the number of heterozygotes present in this. So how many heterozygotes are there? Capital A small a, capital B small b and capital E small e. So totally 3. So 2 raised to 3 is 8. That is the total number of gametes. So out of that you will get only 1. 1 out of 8 will be A, B, C, D, E genotype. So answer is option 2. If the DNA content of the cell in G1 phase of the cell cycle is C, what will be the content after meiosis is completed? It will be half. Meiosis will result in haploid gametes. So it will be half of C. So answer is option 2. Which type of chemical mutagen is incorporated into the genome by DNA polymerase during replication? It is base analogs. Answer is option 2. Then which of the following constitutes a necessary and sufficient condition for two proteins to be considered homologous? So the proteins should have diverged from a common ancestor. That is how you consider the proteins to be homologous. Answer is option 3. Then boiling a mixture of butter, water, eggs result in a homogeneous suspension. However, if the eggs are omitted, then the butter and water separate out. Which component present in the egg is responsible for this behavior? It is lecithin present in the egg. Answer is option 4. Then next one. Cooking meat in the presence of cut pieces of papaya fruit results in unusual tender meat. What enzyme present in the papaya fruit is responsible for this? It is papain. Answer is option 2. Then, which of the following peptide sequences will match the sequence motif? So, they have given a motif there. where G, XX, SVP, XXG. So XX means there can be two amino acids, any two amino acids. The one in a square bracket means either of the three should be there. Not all three, either one of the three should be there. So here they have started with P. Okay. Then G, Q, R. Okay. V. Then again GG and then G and R. Yes, that can be correct. Option 1 is correct. If you see for option 2, they have started with G, then they have taken P. Where it is not P over here, it is G. It should be G in the second place. If it was this one, then G in the second place, then Q and R, then QY. It should be either of SVP. It shouldn't be Q and Y. So 3 is also not correct. First one P, okay, then G, then Q, R, FY. It shouldn't be FY, it is either of SV or P. So the answer is option 1 is correct. Next one. In mass spectroscopy, a quadrupole mass filter does which of the following? So it allows those ions which have specific MZ ratio to pass through. Then next one. If maltose and monosodium glutamate are added to a vinegar and palmitic acid and shaken, the mixture will eventually separate into two phases of different density and polarity. Where will most of the sucrose and MSG be located during phase separation? So both maltose and sucrose and MSG are insoluble in palmitic acid. So they will be concentrated in the vinegar. Okay. So the answer is option 1. Both will concentrate in the vinegar. Answer is option 1. Then a slide of macrophage was stained by immunofluorescence using a monoclonal antibody for TAP1 and TAP2 complex. This complex is found in endoplasmic reticulum. Which of the following intracellular compartments would exhibit positive staining with this antibody? Okay. TAP transporter is found in endoplasmic reticulum lumen. So answer will be option 2. Then which of the following disorder is not X-linked? Color blindness, Rett syndrome, Huntington's muscular dystrophy, Swayer's syndrome. Swayer's syndrome occurs due to mutation of the sex-determining region Y gene on the Y chromosome or deletion of the segment of the Y chromosome. So it is not an X-linked disorder. Answer is option 4. Alpha amanitin inhibits. So it inhibits RNA polymerase. And 2. Answer is option 2. Nullisomy is the term used for the condition when an organism has loss of one homologous pair of chromosome. Nullisomy is 2n minus 2. So loss of one homologous pair of chromosome. Answer is option 3. Then next one. Tunicamycin blocks. So tunicamycin blocks N-linked glycosylation. Answer is option 1. Which of the following cannot be used in finding the interaction between miRNA and mRNA? So it is PAR-CLIP. Photoactivable ribonucleoside enhanced cross-linking and immunoprecipitation. Answer is option 3. All the others, that is target scan, starbase, Miranda, they are all bioinformatics tools. And PAR-CLIP, it is a biochemical method. Okay. Then next one, H1N1, H1N2, H2N1, H3N1, H3N2 are subtypes of which influenza virus? It is influenza A. Answer is option 1. A mother and a father, both cystic fibrosis carriers, have two children that do not suffer from cystic fibrosis. The probability of a third pregnancy producing a child with the disease. So mother and father, they do not suffer from cystic fibrosis. But if they are heterozygous, then there is a chance that 1/4 of the child will be having cystic fibrosis disease. So answer is option 2, that is 1:4. Then next one. The overall reaction catalyzed by the electron transport chain is. So out of this NADH plus H plus half O2 give NAD plus H2O. That is the reaction catalyzed by electron transport chain. Then next one. DNA polymerase 3 of E. coli is. So DNA polymerase 3 is required for de novo synthesis of new strands of DNA. Answer is option 1. Then next one. The protection against smallpox afforded by prior infection with cowpox represents. It represents antigenic cross-reactivity. Answer is option 2. Then next one. Internalized antigens are targeted to which of the following compartment for generation of appropriate peptide for presentation on the cell surface along with MHC molecules. So it is lysosomes. So endocytosed proteins enter a vesicular pathway consisting of progressively more acidic and proteolytically active compartment, classically described as early endosomes, late endosomes and lysosome. So internalized antigens are targeted to which of the following compartments for generation of appropriate peptide for presentation on the cell surface along with MHC molecule. So it is lysosomes. Next one. GTP binding proteins are active in GTP bound form. Which of the following protein convert GTP bound form to GDP form? So it is a transferrin receptor. It is a GTPase activating protein. GTPase activating protein. Answer is option 2. Which enzyme is used to remove the phosphate group from 5' end of the DNA? So it is alkaline phosphatase. Answer is option 3. Then which of the following technique is suitable for large-scale purification of isozymes A and B that are differing from each other by a single positive charged amino acid? So it is chromatofocusing. And chromatofocusing, it combines the advantage of high capacity ion exchange procedure with higher resolution of isoelectric focusing into a single chromatographic focusing procedure. So here they have asked for isozymes A and B that are differing from each other by a single positive charged amino acid. So for large scale purification you can go for chromatofocusing. Answer is option 1. Then glycogen and cellulose are. So it is helical and beta sheet structure respectively. This was also asked for DBT 2013. I have explained it over there. So you can go through that. Answer is option 1. Then which one of the following factors influences the binding of oxygen to hemoglobin? So partial pressure of oxygen. Answer is option 2. Which one of the following DNA viruses has part of its life cycle involving reverse transcriptase enzyme, which is hallmark of retrovirus? So it is hepatitis B virus. It is a non-retroviral virus which uses reverse transcriptase as part of its replication process. Then next one. Cells are broken to release the contents by using various enzymes. Which of the following combination is false? Lysozyme bacteria, cellulase plant cell, chitinase fungus, cellulase bacteria. Cellulase bacteria is not correct. Answer is option 4. Then which of the following enzymes is required to release the tension imposed by uncoiling of strands? So it is DNA gyrase. Answer is option 3. Then next one. DNA fingerprinting is based on the occurrence of variable number of tandem repeats or VNTRs. Answer is option 1. The DNA sequence is ATG. What would be the sequence of bases in anticodon of tRNA? So ATG is the DNA sequence. So mRNA will be complementary to that. You will get UAC, and tRNA will be complementary to mRNA sequence, that is AUG. So the answer here will be option 2, that is AUG. Then all except one of the following are true about protein kinase A pathway. Protein kinase A phosphorylates proteins at serine or threonine residues. That is correct. Protein kinase A phosphorylates protein at tyrosine residues. No, it is at serine or threonine residues. So 2 is the wrong answer. It is activated by CAMP. Yes, it is activated by binding of epinephrine to a transmembrane receptor without enzyme activity. That is also correct. Then a T cytotoxic cell can be induced to mount a cytotoxic attack on a virus infected cell if it binds to a cell displaying MHC1. It is cytotoxic T cell or CD8 plus cell, so it requires a foreign antigen being bound and presented by MHC1 complex. So it is MHC1 bearing foreign antigen. Then testosterone hormone necessary for spermatogenesis is secreted by lady cells. Answer is option 2. Which of the following processes occurs in the formation of disulfide bridge between two cysteine residues? So it is oxidation of sulfahydryl group. Then next one. In Ramachandran plot, the values of the dihedral angle psi is based on the rotation around the C alpha C bond. Answer is option 2. Then next one. Which one of the following antibiotics attaches to 50S ribosome and inhibits peptide transferase activity? It is chloramphenicol. Answer is option 2. The cytological representation of Klinefelter syndrome is 44A XXY. Answer is option 3. Then a chromosome on which T cell receptor alpha chain gene rearrangement has occurred lacks which of the following gene segments? A chromosome on which T cell receptor alpha chain gene rearrangement has occurred lacks which of the following gene segments? So it is diversity. Answer is option 2. Then next one. Which one of the following molecule yields higher amount of free energy? So it is phosphoenol pyruvate out of these. So answer is option 1. Which region of mRNA contains Shine-Dalgarno sequence? The 5' UTR, that is 5' untranslated region. Answer is option 1. Alpha D glucose and beta D glucose are anomous. Answer is option 3. Then which of the following statement is not true of eukaryotic DNA replication? It has multiple origin of replication. Yes. It is synchronized to phases of cell cycle. Yes, it takes place during that phase. It does not involve Okazaki. It involves Okazaki fragments. So 3 is not the true of eukaryotic DNA replication, and that is your answer. The next one. In vitro characterization of E. coli DNA polymerase shows an error rate of 10 raised to minus 6 to 10 raised to minus 7 per base pair. However, in vivo, the observed mutation rate is 10 raised to minus 9 to 10 raised to minus 10 per base. This is all 10 raised to minus 9 is raised to minus 9. Okay, but it is a typographical error. Such discrepancy is because E. coli has mechanism of removing such erroneous incorporation of bases. Actually, they have told that in vitro characterization of E. coli DNA polymerase, it has an error rate of 10 raised to minus 6 to 10 raised to minus 7 per base pair. Whereas under in vivo condition, the mutation rate is very less. Why? So, it is because E. coli has got mechanism for removing such erroneous incorporation of bases. E. coli has got a proofreading mechanism under in vivo condition, but if you do the same process under in vitro condition, you have to provide it with proofreading activity. Only then there will be lesser error rate. Otherwise, the error rate will be more. Then next one. The molecular formula of deoxyribose sugar and ribose sugar respectively are. So it is C5H10O4 and C5H10O5. Answer is option 2. Usually, intracellular pathogens avoid their transport to lysosome for their survival in the host cell. But which of the following intracellular pathogen survives in the lysosome? It is Mycobacterium. Mycobacterium can survive inside the acidic condition of lysosome. It is not Mycobacterium, it is Leishmania. Answer is option 4, not 3. Okay, next one. When HFR strain of E. coli is crossed with F strain, recombinants obtained are rarely F plus because the transfer of the complete F factor rarely take place from when a cross is between HFR and F minus bacterial strain. Next one. Archaea is considered as a separate group from bacteria and eukaryotes based on 16S rRNA gene sequence. Answer is option 2. How many grams of NaCl will be required to make 10 ml of 10 millimolar solution? Molecular weight of NaCl is given. So substitute in this formula, molarity is equal to 1000 Wb by Mb into V. Wb is how many grams you will require of this NaCl. Mb is molecular weight, V is the volume. Substitute and you will get option 3 as the answer. Then next one. Keratin intermediate filaments are synthesized in the cytoplasm of cells. Disulfide bonds cannot be formed in the cytoplasm. However, the keratin fibers in the skin are cross-linked by disulfide bond. This is because option 3. Keratin cross-linking happens in dead cell whose contents are oxidized. Keratin is usually found in dead cells like nails. So in such cells, cross-linking happens in dead cell whose contents are oxidized. Then next one. Which of the following plays a role in changing the antigen binding site of the B cell after antigenic stimulation? It is nothing but somatic hypermutation. It is found only in B cell, not in T cell. Then next one. What region of an mRNA is most commonly associated with transcript destabilization? So what is mRNA destabilization? It is any process that decreases the stability of an mRNA molecule, making it more vulnerable to degradative process. And mRNA destabilization process in which one or more mRNA binding proteins associate with the 3' untranslated region of an mRNA. So answer is option 2. Then next one. Which type of replication requires a break in the nucleotide strand to get started? So it is rolling circle replication. Answer is option 2. Mismatch repair in bacteria distinguishes between old and new strands of DNA on the basis of. So it is on the basis of methylation. The parental strand will have the methyl group, whereas the newly synthesized strand will not have them. So answer is option 4, methyl group on the old strand or the parental strand. The next one. Why did the Environmental Protection Agency closely monitors the release of transgenic bacteria used for agricultural purposes? Because they want to ensure the GMOs do not proliferate in the environment and pose a threat to humans. Answer is option 3. The next one. Which of the following events occur first in the differentiation sequence of human B cells in the bone marrow? So immunoglobulin light chain, no, it is heavy chain rearrangement. It is not light chain, it is heavy chain rearrangement. Answer is option 2. A zooblot helps to detect DNA sequences that. So a zooblot is a type of southern blot that demonstrates the similarity between protein coding DNA sequences of different species. The purpose of zooblot is to detect the conservation of the genes of interest throughout the evolution of different species. So a zooblot helps to detect DNA sequences that are conserved between species. So answer is option 2. The next one. Addition of which of the following can prevent the precocious germination of the embryo during embryo culture? So increased sucrose concentration. So answer is option 2, sucrose. Which of the following techniques can be used to create diploid homozygous plants in a short span, few months to a year? So that is haploid plants derived from anther culture or pollen culture are sterile. These plants contain only one set of chromosomes. By doubling their chromosome number, the plants can be made fertile and the resultant plant will be homozygous diploid. So which technique can be used to create diploid homozygous plant in a short span? That is through anther culture. Then in plant tissue culture experiment, conditioned medium refers to medium in which. So media containing components secreted by cells which are required by another set of cells is called conditioned media. And the answer here is option 3, media in which plant cells have been grown for about 48 hours and the cells are filtered out. So once the plant cells are grown for 48 hours, they will release certain nutrients into this particular media, and this media can be used for growing another set of cells. So media containing components secreted by cells which are required by another set of cells is called conditioned media. So the answer is option 3. An intron containing beta-glucuronidase, Gus intron, is used as a reporter gene to assess plant transformation. The intron is introduced to. So most reporter genes are already active in agrobacterium, thereby preventing the precise determination of timing and localization of tDNA transfer to plant cells. So in order to overcome this obstacle, a portable intron derived from a plant intron was introduced into the beta-glucuronidase gene. In transgenic plant containing this chimeric gene, the intron is spliced efficiently, giving rise to Gus enzymic activity. No Gus activity is detected in agrobacterium containing this construct due to lack of eukaryotic splicing apparatus in prokaryotes. So, an intron containing beta-glucuronidase is used as a reporter gene to assess plant transformation. The intron is introduced to allow, prevent any expression in the bacterial cell. Answer is option 1. Bacterial cell doesn't have eukaryotic splicing machinery, so the intron will not be removed and you will not get colored product after during gas analysis. Next one. Which one of the following is true about epigenetic changes? So changes are caused by DNA methylation and are heritable. Answer is option 4. Then a cross between two breeding lines, one with dark blue flowers and the other with bright white flowers, produces F1 offsprings that are light blue. When the F1 progenies are selfed, a 1:2:1 ratio of dark blue to light blue to white flowers is observed. This is nothing but incomplete dominance. The ratio is 1:2:1 for dark blue, light blue and white flowers. That is, upon crossing dark blue flowers with bright white flowers, you get 1:1, 1:2:1 ratio. It is incomplete dominance. Answer is option 2. In a tissue culture experiment, a student desires to have more differentiation of shoots. Which of the following plant growth hormone ratio should be used? So, high cytokinin to auxin ratio induces shooting in plants. Answer is option 1. The next one. A yeast mutant shows decreased expression of 5.8S rRNA, 5S rRNA, CDC2 mRNA. In which of the following might the mutation lie? So RNA polymerase 1 transcribes ribosomal RNA but not 5S rRNA, which is synthesized by RNA polymerase 3. mRNA is synthesized by RNA polymerase 2. Now Tata box is a DNA sequence to which Tata binding proteins, a transcription factor, binds. Tata box binding protein is required for initiation of transcription by RNA polymerase 1, 2 and 3 from promoters with or without Tata box. So a yeast mutant, it shows decreased expression of 5.8S rRNA, 5S rRNA, CDC2 mRNA. So all these are synthesized by RNA polymerase 1, RNA polymerase 3, RNA polymerase 2. And all these have got a Tata box in the DNA sequence to which Tata binding proteins, that is a transcription factor, it will bind. Only then initiation of transcription by RNA polymerase 1, 2 and 3 will take place. Now the mutation can be, now all these three, that is RNA polymerase 1, 2 and 3 are not able to synthesize all these required RNAs, means that the Tata binding protein itself is mutated. So answer will be option 1. Photosynthesis it is reductive endergonic anabolic process. Answer is option 2. An endergonic process is a process in which energy is required for its occurrence, and in photosynthesis, light energy is used for converting carbon dioxide, water and minerals into oxygen. And anabolic process is a process in which an organism uses energy to form complex substances, and in photosynthesis, light energy is used by plants to convert carbon dioxide and water into glucose. Then in photosynthesis, electrons are transferred from water to carbon dioxide molecule, forming sugar molecules. In this process, water is oxidized as it loses electrons and the carbon dioxide is reduced as it gains electrons. Hence, photosynthesis is a reductive process. So the answer is option 2. Which of the following statement about genomic library is incorrect? So what is genomic library? It is a collection of the total genomic DNA of a single organism. So the genomic library will be representative if they contain all the genes in an organism. Yes. The genomic library must be prepared from cDNA. No. That is not correct. It is not prepared from cDNA. The next one. Which of the following statement correctly describes the sequential steps in cDNA cloning? So reverse transcription of mRNA, second strand synthesis, cDNA end modification, ligation to a vector. That itself is correct. Option 1 is correct. The next one. Hygromycin B, generally used as a selection marker in plant transformation protocols, is. So it is an aminocyclitol antibiotic produced by Streptomyces hygroscopicus. Hygromycin acts by inhibiting polypeptide synthesis. It stabilizes the tRNA ribosomal acceptor site, thereby inhibiting translation. So answer is option 1. So coming to the next question. Which of the following statement about transcription in E. coli is correct? The -10 sequence is always exactly -10 base pair upstream of the transcription start site. -10 region range from 6th to the 12th base preceding the transcription start site. So one option is not correct. The initiation nucleotide is always G. All protein transcripts start with the methionine, which is depicted as AUG codon. The intervening sequence between -35 and -10 sequences is highly conserved. It is not intervening sequence. The sequences CTTGAAA in the -35 region and CCCATNT in the -10 region are highly conserved. Then next one. The distance between -35 and -10 sequence is critical for transcription efficiency. That is correct. So answer is option 4. Next one. In an in vitro culture experiment, the color of explants turned brown just after 24 hours of subculturing. This is due to, so it is due to the release of phenolic compounds from the explant. Answer is option 2. To avoid somaclonal variation in the development of transgenic plant, which one of the following is preferred? So it is direct regeneration. One of the propagation methods of tissue culture is the direct organogenesis. That is, shoot arise directly from the meristematic region without an intervening callus phase. That is called as direct regeneration. Then next one. The transplastomic lines have no risk of gene escape through pollen since the... So a plastomic plant is genetically modified plant in which genes are inactivated, modified, or new foreign genes are inserted into DNA of plastids like the chloroplast instead of the nuclear DNA. A major hurdle of nuclear genetically modified crop is posed by the potential outcrossing of the transgene via pollen movement. Now, plastid transformation yields transplastomic plants in which the pollen does not contain the transgene, and this not only increases biosafety but also facilitates the coexistence of genetically modified conventional and organic agriculture. So the answer here is option 3. Transformed chloroplast DNA is lost during pollen maturation. There is a typographical error here. Transformed chloroplast DNA is lost during pollen M. It is maturation. It has not come. Answer is option 3. And next one. Among the following, which one is not a common method of haploid plant production? The embryo rescue of interspecific crosses, anther culture, ovule culture, colchicine treatment. It is not a method of haploid plant production. It induces polyploidy. Colchicine. Then, which one of the following statements about the M13 bacteriophage is incorrect? It mediates transduction. M13 bacteriophage doesn't mediate transduction. So answer is option 1 itself. Which one of the following are flowering hormones? Florigen and vernalin. Answer is option 2. Which of the following genes are constitutively expressed and control the plant-induced activation of the Vir genes? So Vir A and Vir G. These are constitutively expressed. Answer is option 1. Then, in case of gametophytic incompatibility system, the self-incompatibility phenotype of the pollen is determined by So self or self-related pollen is recognized and specifically inhibited by the female pistil. That is called as self-incompatibility. And self-incompatibility system, it falls into two major classes, that is gametophytic self-incompatibility, in which the phenotype of the pollen is determined by its own haploid genotype, and then comes the sporophytic self-incompatibility, in which the pollen phenotype is determined by the diploid genotype of the parent plant. So here they have asked for gametophytic incompatibility system. So it is haploid genotype of the pollen. Answer is option 1. Then high frequency heterokaryon formation is observed during protoplast fusion by the addition of PEG or polyethylene glycol. Answer is option 2. Somatic embryo induction generally occurs in the presence of the two growth regulators, namely, so it is auxin and abscisic acid. Answer is option 2. Then next one. A molecular biology student genetically engineered Arabidopsis thaliana to harbor BAR gene. The resulting transgenic plant is expected to be resistant to. So it is BAR gene introduction. It is resistant to DL phosphinothresin. Answer is option 1. Okay, next question. Which of the following statements about haploids is incorrect? They have help in shortening of breeding cycle. Using haploids, it is possible to obtain exclusively male plants in dioecious species. Yes. Haploids are useful in isolation and detection of mutants. Yes. Haploid plants cannot be used for gene transfer. That is wrong. They can be used for gene transfer. So option 4 is incorrect, and that is the answer. Then refugia approach in cultivation of BT cotton is to minimize. So it has been observed that BT cotton is getting affected by bollworm, which is typical case of BT resistance. And the self-mated progenies of this resistant insect lead to further generation of resistant insect population. So it is necessary to plant 20% refugia crop, that is non-BT cotton, around this BT cotton for maintenance of BT cotton resistance against the bollworm. So refugia approach in cultivation of BT cotton is used to minimize emergence of virulent biotypes. Answer is option 4. In which method of plant transformation would multi-copy integration be a common feature? So it is particle bombardment. Answer is option 1. Which one of the following techniques will help to overcome a pre-fertilization barrier between two species? So it is protoplast fusion. Answer is option 2. Genes located in which one of the following do not follow Mendel's law? So it is nucleus, no, chloroplast, yes, and mitochondria, yes. So both 2 and 3. Answer is option 3. They do not follow Mendel's laws. Seedless fruit can arise as a result of parthenocarpy. Fruit developed without fertilization in the case of parthenocarpy. So there are no seeds. So seedless fruits can arise as a result of parthenocarpy.